Okay, guys, good morning, everybody. I wanted to start with this article that I came across yesterday, I think it was. Sometimes I find these and I'm like, oh, and I email it to myself. And I thought this is super fitting to what we had talked about with the first chunk of material. Unfortunately, it posted after we already dealt with it, but I wanted to share it with you. I'm not going to go through much detail with it. I did post it on Moodle, the link, so you can access this. This is coming out of New Atlas, but it was from the journal Science originally, and there, I think at the tail end of it is a link to that. So real quickly, before I even think about that, I would like to mention a little bit about journals and things like that. Science is something that we would call the primary literature, where scientists write their articles, they publish them there after they have been evaluated and reviewed by other scientists. All right, that's the primary literature. A secondary source would be something like New Atlas or Time Magazine or whatever you might be getting your information from. And ultimately, in those situations, they take that information and kind of distill it down into maybe a little bit more understandable terminologies. All right. So if you truly are interested and want the full facts, you can go to Science and see that. So if you recall, starch is a substance that plants will produce when they are storing up glucose molecules. Our bodies store up glycogen. It's same but different, right? This is glucose or sugar storage molecule, a macromolecule, right? So plants do this, and they do it pretty stinking well, all right? Or at least so I thought, all right? There with this article is artificial synthesis of starch from thin air, if you will, from CO2. And it apparently is much faster, much more efficient than plants. And there are numerous things that I think about with that. So starches are used in all sorts of stuff, all right? More than just things that we consume, like in a potato, all right? And for industrial stuff, this is huge, all right? So they've gone through and talked about a handful of different things in here. And they use, like how I showed before with the metabolic pathways, all of those different steps governed by different types of enzymes that help to fuel these reactions, right? So somewhere in here it talks about a catalyst, and recall that is an enzyme, right? It's a protein, and they have a normal occurring protein that starts this first bit, this organic catalyst, and it takes the CO2 and produces methanol, a type of alcohol, right? Not something you want to consume, right? From that, from the methanol, they in turn use some engineered enzymes, some things, some proteins that are going to manipulate the methanol molecules and convert them into sugars, into starches. Really pretty impressive, right? So it, as it goes on to say, 8.5 times more efficient than plants like corn, where we would be thinking about getting lots of the starch in the first place. I thought about that a little bit as I was driving to school today. I was like, you know, that kind of makes sense because the plant, that corn growing out in the field, isn't just pumping out starch. It's using these CO2 molecules that they're pulling in for all sorts of other things than just starch production. And so that ultimately is a huge factor for why it's so much more efficient in this reactor, in this lab situation. we're not using the CO2 for growth. We're not using it for other things. We're just using it to pump out starch. And so you get a considerably higher yield when all you're doing is building starch, right? That's pretty impressive, right? So much less space. It says in here, a cubic meter bioreactor. These are just big, like vats that are allowing these reactions to occur. Piping CO2 into the mix and getting starch as an end product. There are all sorts of reactors, bioreactors, where we use like bacteria to produce different things. This is a little bit different, right? A cubic meter reactor gives us this output that is about the same as a third of a hectare. And a hectare, that I'm not 100% with the size of that. I should probably know those things, but I don't. But really pretty impressive. So again, if you are interested, you can access the full stuff. But pretty cool. Okay, I'm gonna stop that one. I do recommend you kind of just take a peek at it because it is pretty interesting to think about some of those potentials and the ability to, from starch molecules, we can manipulate all sorts of things and produce numerous other molecules. So it really sets the stage for all sorts of possibilities with industrial applications. Okay, so this chapter, this is the last before the exam. There are a handful of slides. My thoughts are probably going to be kind of like the last one, where we'll lecture today, we'll lecture Tuesday, and then Thursday we won't have class. You'll have time. You will have multiple days to deal with it, but you can, if you want, you can deal with it on Thursday. Is that good? Anybody's like, no, man, I really want to be here the whole time. Pretty sure that's not the case. Okay, so wait a minute. Let's get this going for real. The heck am I doing? All right, now we're ready. So this chapter, we start thinking about some details with the DNA. And we have already talked about some of this stuff, the littlest bit, as we talked about chromosomes. And you get 2 copies, one from each parent, contributing to your entire makeup. So you get 46 total chromosomes, 23 from each, all right? If we're talking ourselves, all right? And so when we think about that, there's all sorts of possibilities. We've talked about genes that are dominant and genes that are recessive, but we haven't really thought a whole lot about that yet. And that's what we want to think about today. We want to think about how some of these things interact, how they're potentially passed on from parent to offspring. And with that said, we can think about some of the phenotypes, what we would think of as the outward appearance, right? Based off of a specific genetic combination, we might be able to visualize specific things, right? So to start with, when we start thinking about inheritance, we think really genetics, right? And with that, we'll come back to the P. I want to start with this side. I should have rearranged them the littlest bit. Gregor Mendel is the individual that we credit with coming up with these ideas, these concepts. We think of him as the father of genetics, all right? And I will back it up to the pea plant now. He studied genetics and inheritance with pea plants, right? And there are all sorts of variations that we can see. Sometimes, maybe you've planted a garden, planted some peas, and you might see a little bit of variation just in, that row of peas that are growing, right? each pea plant potentially a little bit different, right? Maybe some flowers are different, right? It kind of depends, right? The size, the shape, the peas in the pod, all sorts of possibilities, all right? Variations that you might see, all right? So he uses these pea plants and he grows thousands of different types of peas, plants, and harvests the peas and saves them and plants them and figures out what sorts of things are happening, all right? So let me go to the next slide and back it up one little bit before we start planting these peas, all right? Gregor Mendel was a monk. He was an Austrian monk, as we can see from this image. He a lot of his schooling that he had kind of drove him to where we're at as we think about this genetics, right? He was hoping to become a teacher. That's what his end goal truly was. As he went to school, he studied mathematics. That's what he really liked. He liked all sorts of numbers, all right? Statistics and probabilities, all right? Details that in the biology realm, we don't want to think about too much, all right? Sometimes that stuff, we just kind of clash a little bit, all right? So there are also things that we're thinking about at the time as far as how things might be inherited in the first place. How is it that these things come about, right? There were thoughts that when a baby is born, it was the result of a little preformed human being in the sperm cell of the male, and upon fertilization, that's what you get, right? That seems ridiculous, right? We recognize now that's not the case. And I would like to think, well, what would happen when that child hatches out and looks just like the mother and nothing like the father? How do you explain that? How do you write that off, right? Those are definitely some issues. And I think it made people start to wonder a little bit about some of these ideas, right? We know that doesn't make sense, all right? Who came up with that idea? Some dude, all right? Must have, right? So that's beside the point. There were other thoughts besides the preformed human in the sperm cell, thoughts that upon fertilization, there's the shuffling of the genes. And once they're shuffled together, they never come back out of there, all right? And that's also not true, all right? And Mendel starts thinking about some of these ideas, all right? So doing this app, like I jumped ahead from his schooling. So he takes all of these courses in math. That's what he wants to do is teach this stuff. But as he is trying to become an instructor, there were issues with, whatever it was, exams, and he wasn't quite up to par with some of the other materials that would make him, I guess, prepared, I suppose, all right? So he didn't pass the exams, right? But he's still focused on some of these ideas, right? So he goes on into the monastery, and that's what he ultimately does. That's his calling, I suppose. He becomes a monk and still keeps these kind of thoughts about math, right? And when you're thinking statistics, when you're thinking about some of these things, you need numbers. And the more numbers, the better. You need data, all right? And there are all sorts of ways you could come about this. And he, fortunately, used a biological concept. He used some biology to generate data. And he generated massive amounts of data from all of these pea plants that he grew, thousands of pea plants, right? So for statistics, that's what you want, data, all right? So he wasn't really a biologist, but he definitely did biology, right? And he got massive amounts of data. And then that's what, ultimately what he wanted, all of these numbers, right? What does it all mean, all right? So we will get to this in a second, all right? I also want to point out, all right, we have shown pictures of chromosomes. We have talked about these things. We've talked about a little bit about genes on these chromosomes, the littlest bit, all right? He didn't know any of that. He had never seen a chromosome through the microscope. He hadn't seen any of these things. He talks about some of these characteristics, the traits in these pea plants, as particles contained within this seed of the pea. That particle would be a gene on a chromosome, right? But again, he hasn't visualized any of this stuff, right? Did you guys look at onion root tips and chromosomes? Have you seen that stuff? Okay, so you've seen it. You've already seen that stuff. He never had any idea what that was, right? So keep that kind of in the back of your mind, all right? He's kind of starting from scratch, if you will, all right? So in order for Mendel to evaluate these things, these characteristics or traits observed in the pea plants, he had to ensure that what he was working with was pure stock, if you will. It was always going to give him that same stuff, all right? So he creates these what we call true breeding plants, or a true breeding pea plant. The first thing we're looking at, and there are a handful of different traits, all right? So each one of the specific traits, he did the same thing and made sure I always get the same thing, all right? With pea plants, and then, plants, there's all sorts of potentials, there's variation. Some plants can cross-pollinate, and they have to cross-pollinate in order to create viable seeds. And other plants can be cross-pollinated or can self-pollinate, and they can create viable seeds, right? Pea plants are that way. They can either cross or self-pollinate, and you can control that by manipulating the flower, all right? So initially, he allows the violet or the purple flowered pea plants, he allows these guys to grow. He pollinates, self-pollinates the pea plant, collects those seeds, plants them, grows them, purple plant again, pollinates, plants, and grows. He does that a handful of times and ensures every stinking time it's a purple plant, right? And he does the exact same thing with the white flower plants, all right? These are true breeding plants, right? And what we will say here shortly is these are homozygous, two of the same copies, right? And ultimately, one of them is going to be dominant, two capital letters. That's how we express dominance, with capitals. And the other one will be recessive, two lowercase letters, right? We don't know which is which at this point. And with the pea plants, Mendel had no clue. He figured it out, right? So as this happens, right, he gets each of these true breeding plants, and he ensures that's what you see is what you get. Now the question is, what happens when we allow a white and a purple to cross-pollinate, right? As this occurs and he mixes, and he did both ways. He did pollination onto the purple plant or pollination onto the white plant from the opposite colored plant, just to see what's happening, right? Plants have pollen and ovules, there's pistils and stamens. We'll see, I think, a little anatomy of a flower. We don't need to get super bogged down with that, all right? But with that said, he could control and could manipulate these things. So he allows these guys to pollinate, vice versa, and grows up, they grow up, produce the peas in the pods, he harvests those and plants them. When he plants them, the F1 generation means the first filial, these are the children, all right, of this mating, all right? And each of these plants produce the peas on the pods, he harvests them, plants them, and these plants grow up. Every single plant that every single pea that he planted grew up to have purple flowers, all right? Even though there were white flowers in the mix, all right? So what's happening here is this hybrid progeny, right? This mixture of the two, and they are all purple, all right? Initially, as we see this, this looks like this shuffling, this blending, right? They're all mixed together, right? And they're all purple, right? These guys, we will call here shortly heterozygous, right? One big allele or copy and one small gene or allele, all right? And an allele is just an alternative version of that gene. So the allele for flower color, you might be purple or you might be white, right? Whatever other plant, you could have a whole variety of different possibilities, all right? So now these purple plants, the children of this first parental breeding, these peas are allowed to self-pollinate, right? So there's no crossing going on. It's this plant pollinating itself, right? It's got its own genes within it. He lets them produce the peas, collects them, plants them, and the F2, well, these are the grandchildren of these guys, the grand peas, if you will. The F2 generation, when these guys grow up, all of the sudden, here's some of the white ones. You don't see them in the F1s, but you definitely do in the next generation, right? In the grandchildren, right? You probably have heard things, maybe parents telling you something, maybe somebody saying, oh, you know, grandpa had that, or your grandmother had that, something like that. Things skipping a generation, right? That's what you're looking at, right? You're looking at some of these things that you may not see in your parents, but you do see in yourself, right? And if you look at your grandparents, if you happen to know them, you might see the same similar sorts of characteristics or traits, all right? It really kind of depends, and that's really pretty oversimplified, but it is kind of what we're thinking about right here, all right? So this is just one little bit, but it's a massive amount of data that he's already acquired as we look down here from this next generation. So handfuls of these pea plants grown up, creating peas in the pods. Typically a little pea pod has 4 or 5 peas inside of it, each of which could have some variation within that, right? And as he plants them up, now we've got 705 pea plants in these F2 generations that all have purple flowers. And we've got 224 that have white flowers, almost 1,000 pea plants, all right? And I think now the littlest bit about the monastery and these poor monks, as Mendel's out in his garden growing boatloads of peas. How much split pea soup do you think those suckers had to eat? Every day, right? And that is gross. All right, maybe not, maybe that's your favorite kind of soup. So right now, first get my pointer back here. At this point in this F2 generation, we can see that what we thought of as this shuffling, it goes away, you never get it back. It's not true, because the second generation, the F2s, it shows through, all right? So the dominant characteristic with the pea flower color, the dominant trait was purple, and the recessive trait was white. And when you mix a dominant and a recessive, they are both homozygous, right? Homozygous dominant, homozygous recessive. And when you mix those two together, all you get are heterozygotes, one dominant and one recessive, and the dominant prevails, right? So purple, in this case, prevailed. That's how we know it's dominant. And we know it because Mendel did all of these things in the first place and figured out naming stuff, dominant and recessive and so on, all right? And here we can see over the course of these generations, the next generation here and these plants, we've got a good chunk of them that still exhibit that dominant trait, and some over here, the recessive, all right? With that said, we know the genotype of these parents up here, right? we will say capital P because the purple is dominant. 2 big P's and you are purple, all right? 2 little p's because we keep the same letters, the dominant letters, what we use, all right? So 2 big P's and you're purple, 2 little p's and you're white, all right? Down here, we're a big and a little, all right? 1 big P, 1 little p, and we're purple, all right? This one, we know, all right? We know that white flower pea plant has to have 2 little p's, right? But when you look at these pea plants, can you tell me what their genotypes are, what their specific letters are? You can't, right? There's a good chunk of those pea plants that are gonna have 2 big P's. But there's also some of them in there that are gonna have a big P and a little p. And you can't tell just by looking at them what they have, right? But with the recessive, you can, all right? So we can show some more of that, and we will see some more details with how we can make some calculations and figure out some of those potentials. And really, there's a bigger chance to be heterozygous, 2 copies, than just homozygous. So as I have mentioned, he looked at all sorts of things, all right? More than just the flower colors, all right? Pea plants do have a lot of variation within them. And sometimes you can see a handful of these same things on the same plant, all right? But when he initially starts this stuff, he takes one specific thing and he makes sure it's true breeding, and then he evaluates, right? He takes it a step further and starts looking at what happens when we look at multiple things together. And that makes it even more data points, right? It makes it even more fun for his mathematical mind, all right? So we showed the flower colors, which we've got right here in the middle. The shape of the peas, the seeds, can be round or they can be wrinkled, all right? And as it turns out, as we are showing this stuff right here on this slide, the dominant stuff is all on this side and the recessive stuff is on the other side, all right? And again, because this is how he figured it out, doing these crosses, all right? So seed shape could be round or wrinkled. The color could be yellow or green. We've already seen the flowers. The pods themselves can be inflated, puffed up, or constricted, sandwiched around those peas. The pods themselves can also be colored yellow or green, all right? And there are definitely potentials that you may see some of this variation if you plant a whole packet of seeds into the garden. Even just putting them into your hand to plant them, you might recognize some of them, probably the bulk of them, are round, and then a handful of them are kind of wrinkled up, all right? And this is all when they're dried, all right? You're not gonna open up the pea pod and see a little pea in there that's all kind of wrinkled looking. This is a little bit later, all right, as it's dried. I believe, I don't think it looks like that right off the bat. They're all round. This is after it's had some time to dry. So 7 different characteristics. And like I said, each separately, and then he starts thinking, well, what if we mix it up? What if we have the 2 together, or 3, or all 7, all right? We won't show you all 7 because that becomes too chaotic, right? He also looks at I skipped over these guys, but this is part of it as well. The flower position on the plants, it can be axial or terminal. And then the plants themselves might be great big. They might be tall pea plants, or they could be little dwarfs, right? And again, looking at all of these specifics, all right? So I mentioned a little of this, and I mentioned genotype today, just a few minutes ago, all right? The phenotype's the outward appearance, right? And in this case, the color of those peas, right? The genotype are the specific chromosomes, those genes on those chromosomes, I should say. And as we indicated a second ago, the recessive stuff, well, it's easy to pick out, right? Once we look at the others, it's a little bit more difficult to figure out, all right? So with this, with the color. So the P again are the parental populations. We've got true breeding yellow peas, true breeding green peas. We cross fertilize these guys, and every one of the peas that grow up in these plants are yellow, all right? Hybrids, right? They are heterozygous, right? A big Y and a little y, all right? Because they're all yellow here, all right? Allowing this F1 generation to self-pollinate gives us this ratio. And 3 of the 4 are yellow, and 1 of them is green, all right? So a 25% chance of having this recessive characteristic, right? And a 75% chance of having the yellow, right? These are all probabilities. It's coin flips, right? There could be a pea pod that you cracked open and every one of those suckers were green, right? It's super unlikely, but it could happen, all right? It also could be every one of them yellow in this pod, all right? If you had 4 peas in that pod, all right? There could be variation, all right? There should be variation, and not every single pea pod that's cracked open is going to be 3 yellows and 1 green, all right? Doesn't happen like that, all right? But it does show you some potentials, the odds, all right? We could think about children, right? Humans, right? Female 2 X chromosomes, a male an X and a Y, and upon fertilization, each donates one of those chromosomes. The female's only donating X's. The male could donate an X or a Y, all right? So now, upon the fusion of the egg and sperm, you've got 50-50 odds of having a boy or a girl, right? Dependent upon which chromosome the male donated, right? 50-50 odds, all right? But that doesn't mean you have 2 children and one's a boy and a girl, right? It could be, right? But maybe they're both boys. Maybe they're both girls, right? 50-50 odd doesn't mean you get one of each, right? It's probabilities, right? So, and with that, data is important, right? Having massive amounts of data points helps to get to these conclusions, right? Sampling 1 pea plant, I don't think you could be drawing these conclusions, right? From looking at a handful of peas coming off of this pea plant, right? But sampling hundreds or thousands of pea plants and then collecting all of that data, that's where you start getting this information, right? So the phenotype is the outward, the visible characteristics that we can see, and the genotype is the genetic makeup, all right? So here we can see with these fertilizations, and really it should be flip-flopped around, all right? Big Y's and little y's if we're keeping it consistent to this, because yellow is the dominant, all right? These would be your green ones, and these would be the yellow ones, all right? So 50 or 100% over here, big Y, little y, all right? It's half of each, all right? But the dominant gene, that's what prevails in this type of inheritance, all right? And then we see down below, all right, through self-fertilization and the potentials that we could get with this if we were to actually figure out the specific genetics behind it. 25% of these yellow pea plants are going to be dominant, homozygous dominant. 50% will be like this, right? There's 2 ways to make heterozygous, right? Dependent upon which one, right? And then down below, we've got 25% that are green. There are ways we can go about this. Punnett squares, we'll see some of this in just a second, where we can move through and think about how these are being passed on. Are you guys doing that in the lab? Is that happening right now? Punnett square, did you do any of that? Next week? Okay. So there are a few things that we need to think about that Mendel truly brought to biology, all right? And this stuff, it didn't happen overnight, right? Mendel, as he starts getting all of this data, compiling it together and presenting it, like I showed the journal Science, all right? It wasn't the journal Science, but the article that initially was in the journal Science, all right? If you see one of those journals, there are all sorts of different disciplines that publish within that, right? And if you are studying biology, you highly unlikely are going to be reading an article about quantum physics, right? I'm pretty sure you're not, right? You're going to pass that right over, right? So Mendel starts making some of these, publications. He publishes some of this research, and it's all mathematics. It's full of statistics. It's full of all of these numbers. Biologists don't look at any of that, even though he's talking about stuff that fits in biology. It's all math, right? And so it gets kind of brushed to the side. No one really looks at it, or if they do read it, they don't pay it much attention because they get bogged down with all of the math, right? So he kind of gets forgotten, right? I kind of like to think of him like, the starving artist that gets all of these works, then they die, and now their stuff is priceless, right? They didn't live to see the benefits of that. That's kind of what happened to him. It was about a decade or so after Mendel had passed away that his works were rediscovered by a biologist, that as they blow the dust off of this manuscript, they're just like, what in the world? It all makes sense, right? So kind of crazy, right? He comes up with a couple of laws that we still talk about, we use them, we think about them, but in biology, we don't think about laws. We think about hypotheses, we think about theories. We don't think about laws because that is in the realm of mathematics. There are laws. If you were a mathematician, you would say it's the purest of all sciences, right? Stinking buttholes, right? They don't know, right? They do. It's true, right? You can prove these things. You've got all of these theorems and proofs that justify that. You can show it, all right? Would it never prove a theory to be 100% true? There is the potential that there could be some flexibility with that. It's for what we think about, it's pretty much 100% true, right? But there is in biology the potential for some change, right? So these laws make it a little bit different, right? And ultimately, we talk about it because Mendel wasn't a biologist; he was a mathematician, right? And if we flipped it around the other way, we would be talking about these as theories. If it was truly a biologist that had postulated this in the first place, it would be a theory of dominance or independent assortment, as we will see in a minute, all right? So that's all fine and good, all right? Just keep that kind of in the back of your mind, the littlest bit. That's why we see some laws, right? So the genotype and phenotype with these specific dominant and recessive situations, right? This law of dominance states that a heterozygote, one that's got both big and little copy, the dominant will prevail and the recessive is masked or hidden by that dominant trait, right? And that's what we can see, maybe, in this table. So the homozygous with the two dominant, the heterozygous with one of each, both yellow, right? Prevailing over the green with two recessive alleles, right? So the law of dominance, the big copy, if you will prevail over the small. Sometimes, as we think about this, as I talk about it, I think of good copies and bad copies, dominant and recessive, but that's not 100% accurate, all right? Two dominant characteristics may be good, they may be bad, right? The same with the recessive, right? When we think about some specific genetic disorders, Lots of times they are recessive things, but again, not always, all right? There are a lot of different potentials with that, all right? So one, just as we're mentioning that real quick, if you take a peek at your fingers, right? I bet you every single one of you in here has 10 digits, right? If you took your shoes and socks off, but don't, if you did, I bet you've got 10 toes, right? Probably. Anybody deviate from that? Silence. How about anybody tuning in from home? Any extra fingers, pinky, thumb? Nobody says it, all right? Most of us would say we've got 10 digits, right? Having 10 digits is a recessive condition, right? That's the norm, all right? So recessive, like I said, recessive isn't always bad, all right? And it's not always the most uncommon, all right? In this case, the recessive trait, normal digits, is the norm, all right? Polydactyly, having extra digits, is a dominant characteristic. You only need one gene and you got an extra thumb, right? Or pinky or whatever, all right? But it's not the norm, all right? So keep that in mind as well. Just because something's dominant doesn't mean that it dominates and becomes the most prevalent, all right? That's not the same definition, all right? And the same with the recessive, all right? So I mentioned before albinism, and that is a phenotype that you can tell, all right? And it is an autosomal recessive to, well, little y's in this case, all right? Two little a's for albinism, right? And in turn, specific characteristics, no pigmentation at all right? I think the eyes are really a light color as well. It's not like, I think of like albino rabbits with pink eyes. That's not the same with humans. I don't think their eyes are pinkish like that, right? I think they are very, very light, but not pink, right? But hair is very blonde, skin color is unpigmented, and in situations like this, definitely some sort of oddity, I am sure, right? You know, the first albino that, you know, happens in a situation like this, you're like, what the heck, right? How did this happen, right? There are other ideas like, you know, a deer or an elk that's albino. You don't see a whole lot of them, right? They are there, right? But you don't see a tremendous amount. If you see a herd of deer and there is an albino in that herd, there must be some heterozygotes within that population in order for that albino to have been created in the first place, right? If there is a herd of deer and they all look normal colored, you can't say for sure, right? Are they all homozygous or is there some heterozygotes in them? There's probably some heterozygotes, but you can't tell because they all look the same, right? If you see some difference, there you go. And I assume this little dude's sitting on its mother's lap. So if that's the case, we will say she obviously is heterozygous, one dominant and one recessive allele, all right? And if the father was on the other side over here and he was pigmented as well, he would also be heterozygous, right? One dominant and one recessive. But just looking at them, you couldn't tell what their genotype was, right? Their phenotype is pigmented and his is not, all right? I'm assuming that's a guy, all right? So both parents had to have carried a recessive allele, all right? Unless the father that we're not seeing, right, is also an albino, then he had two recessive alleles, right? And there you go. But you have to have two in order to have that happen, right? This was years back. We went to the Hogle Zoo in Utah. We took our kids down there. We were walking around and we got to the alligator exhibit. And there are all sorts of regular alligators, green alligators. And as we kept kind of looking at them, down kind of right beside the wall is an albino. And very odd, right? All of the regular alligators, I had no issues with, seemed all fine and good. And then I look down and see this just white alligator with pink eyes, and it's just staring right up at me. And I felt very freaked out. It just seemed wrong. And I felt like it was just trying to get me, like hypnotize me to come into the pen so it could eat me. I just knew it, right? I'm like, this thing, it's trying to possess me. Odd. And it wasn't, right? But it was definitely odd to think about, right? So maybe in a situation like that, maybe that albino alligator is something that the zoo would like to have more of, right? If you've got one of them, then there must be some parents that are heterozygous, because everybody else in there was green, right? There must be some that are heterozygous that in turn could create some eggs in that clutch that are albino, right? And we could ultimately figure that out, right? With people, technically we could do some of these crosses to figure it out, but that becomes considerably more difficult, right? And it takes a long time for a child to be born to become of reproductive age, and thinking about crossing them with just some known individual, that's kind of an issue, all right? We'll touch a little on that in a minute. So second law, we've already looked at this in the last chapter, right? When we were looking at meiosis and how these chromosomes start to separate. The law of segregation is how these chromosomes, the homologous pairs, when we're going to come together. So we've replicated and generated sister chromatids, and then we bring them together. And we're thinking paternal and maternal as they're coming together, and then they can be different as they're assorting along this metaphase plane, right? As we start to line them up, pairing them up, here you can see reds and blues, and then blues and reds, right? Different, right? That variation, if you've got 20, if we're talking ourselves with 23 pairs of chromosomes, the way that these guys all line up in each individual egg or sperm cell, that's going to vary. It's random, right? And if you happen to have siblings, you see some resemblance, but you most likely see some variation as well, all right? Differences as the result of specific combinations of chromosomes that you potentially got, right? It's random, right? And this law of segregation basically shows that, right? You've got numerous potentials for these things to occur, all right? So I don't remember the exact numbers, all right? But you guys have probably heard that, talking about how special or how unique you are, you're one in a million, right? You've probably heard that term, right? And if you haven't, well, you should have, right? Everybody, right, is pretty unique. Unless you're an identical twin, then you're not so unique, right? But you're still pretty unique, right? And if you happen to be identical twins, well, then I think you're, that's another step up, right? So 1 in a million seems like that's pretty, right? Pretty unique, pretty special, right? But that's not even the tip of the iceberg when we think about how unique we truly are, right? With all of these 23 pairs of chromosomes and the randomness, this law of segregation for how they are pairing up along this metaphase 1 first round of meiosis, all of the randomness with that, with the 23 different potentials, and then taking it a step further with the randomness of the specific egg that is ovulated and the specific sperm cell that ultimately is the one that does the fertilization, brings you more from 1 in a million to somewhere around 1 in 64 trillion. And now you can sit there and say, good hell, I am unique, right? I'm pretty stinking special, all right? 1 in 64 trillion is definitely pretty crazy for the amount of assortment within that, all right? You've also probably heard, you somewhere out there, you've got somebody that, a twin, right? Maybe, maybe not, right? Then you're not so unique, right? I don't know, it's odd, right? There are definitely some images of, children, offspring. The Kennedys are one that's often put into some biology books because you see all of the Kennedy brothers standing together and it's just like based off of size, but they all look exactly the same, right? Kind of odd. You might say, oh, those are good genes, or maybe it's just specific things that are expressed, something that's, each of them had, and in turn, those specific traits. I've got two boys. Both of them looked very similar, and when they're a handful of years apart, very, very similar. There are definitely some differences now as they're getting older, but initially very similar, right? And again, it is random, right? So back to that albino alligator, right? Maybe we want to make some more of them. How do we know, right? In order to determine some of those things, you can do a cross, a test cross. And that is where we take a known, so a recessive, the albino, or in this case, the little green, little Y's, the green pea, and we question the other plant or the other alligator, right? And could this one, it's yellow, right? So we know that it's got at least one dominant allele, but does it have two dominant or is it heterozygous, right? So by mating these two together, you get your potentials, and this is this or that, right? If it happens that it is dominant, right, homozygous dominant, two big Ys, then every single thing that we get, the only way you could have anything is being heterozygous, you're all yellow. If it happens that this yellow one is a heterozygote, so a little Y over here, now we've got 50-50 odds, half of them yellow and half of them green, right? So we could take that albino alligator and we could mate it with a green alligator, right? We don't know what that green alligator is, but maybe it's heterozygous. And if it is, I don't know how many eggs an alligator has, but half a dozen at least, I would think, right? And if a few of those eggs or even one of those eggs hatches out and we've got an albino, it's a done deal and all right? It is still probabilities, all right? Could you have a heterozygote alligator that mates with this albino and they all look green? For sure, you could, and you wouldn't realize that. But the odds, especially if you have multiple offspring, are more in your favor of pitching that recessive situation. If we're talking humans, that's a bigger issue, all right? And you may have a handful of offspring and never even recognize it, all right? Cystic fibrosis is a recessive condition, two bad copies, if you will, all right? And cystic fibrosis is bad news, right? You've got super thick mucus within your lungs, within your GI tract. You've got all sorts of infectious disease and malabsorption issues. I think there are some good treatments now, and life expectancy is definitely getting pushed upward. But back in the day, you were lucky to make it to about 20, all right? Bad news, right? So parents of a cystic fibrosis patient, these parents are normal, right? But they're heterozygous. They've got a dominant and a recessive copy, right? And with that said, it's 25% chance that they create a child with cystic fibrosis. 1 in 4, if you will, all right? But you could have 4 kids and they'd all be normal, right? You could have 4 kids and they all have cystic fibrosis. I bet you don't, right? I bet you if your first child has cystic fibrosis, you probably think we're done, right? Maybe, maybe not, right? Depends on how much of a gambler you are, right? 75% odd, those are pretty good odds that you would have a normal child, all right? But if your first one has that, that might be the end, all right? Truly kind of depends. Depends on your gambling, right? So numerous ways that we can evaluate these things and test for specific. When we think about people, we don't really think about this as a way to figure out what's going on, right? You don't just randomly breed with somebody, right? Probably not, right? So the Punnett square, and with this, Punnett comes up with a way to visualize the statistics, the data, all right? Mendel wasn't doing Punnett squares. He wasn't making these boxes and he was doing math. He was calculating the statistics, the details. What are the odds of this happening, all right? And for a biologist, I already mentioned this. We get bogged down with that. We're like, why, right? Why all of the detail like that? We don't want all those numbers, right? We're good at collecting all of it, evaluating it. Oftentimes, biologists befriend somebody in the math department. They like us because we give them data, and we like them because they crunch all the numbers, and it works together, right? So Punnett is a biologist, not a mathematician, and he thinks, how in the hell can we make heads or tails of all of these numbers? I want to visualize it. I want to draw a picture, right? I want to see it. I don't want to calculate it, all right? And it makes it a lot easier for us to wrap our heads around, I think, all right? To just see what's happening, all right? So it gives us a visual, all right? So in a monohybrid cross, it's one thing that we're crossing, right? One trait. In this case, the P color, right? We started the lecture with a monohybrid cross with the purple and white flowers, right? Just testing one trait, all right? So that's all we're looking at here, yellow versus green. And the genes, the copies of those chromosomes are expressed inside of the P, right? One big allele for Y, another big allele for Y, we got a yellow plant. Two little alleles that are lower Y in this case gives us the green, right? So the gametes, the egg or the sperm, ovules or pollen if we're talking plants, but same idea basically, are brought down below, all right? And here we're only showing you one, right? But there's only one chance here, right? Of these, you're only gonna get a big one and you're only gonna get a little one, right? That's all you've got, right? So the F1 gives us the heterozygote, big copy, little copy, and then the gametes from it are one of each, right? And as we bring it down below, the Punnett square, for a monohybrid cross, it's a 4-window pane square, and we put our alleles on the top and on the side, all right? So with this heterozygote, we know we've got a dominant and a recessive, right? And we put both on there because we're going to allow it to self-pollinate, right? So this is the only chances we've got. As we bring them down, we bring our letters down, we bring our letters over, and we fill them in, right? So this and that gives us the homozygous dominant, the big one and the little one, homozygous recessive, and the little and the big, another homozygous recessive. When we do this, we show the dominant in the front and the recessive in the back. But as we look at this, if we're dragging them down, we would have the dominants here and we have the recessives on the front. And so you could have a capital Y, lowercase y, or a lowercase y, capital Y. 2 ways to make a heterozygote, all right? 50% chance of being heterozygous, 25% chance of being homozygous dominant, and a 25% chance of being homozygous recessive, right? And down below, we could get the ratios. So this is all based off of the phenotype, the outward appearance. 3 of them are yellow and 1 of them's green, a 3-to-1 ratio, right? 75%, 25%, right? And Mendel, growing all of these stinking pea plants, has hundreds or thousands of data points for each of these. And he goes through and calculates it, and it's not exactly 3-to-1 every time, but it's pretty stinking close, right? 2.97 or something like that. It's very close, right? To 3-to-1 odds, right? And as I mentioned, as we were kind of starting this, if it was just one pea plant, you're not going to be able to get that sort of information, right? It's not going to get it to you. Okay. I don't know where I'm at with this, but I feel like it's probably a breaking point. You guys have questions or concerns? I think we should stop right here. We'll pick up with the rest of this, the dihybrid, mixing 2 together on Tuesday. All right, guys, have a great weekend.